My name's Honora O'Neill. I'm a philosopher. I used to be a Gates trustee, and it's a terrific outfit. And we have a panel on some interesting, no sooner do you start than everybody remembers that it was starting at 4. But we have a panel on some very interesting topics, but just to set your minds at rest, we won't try to cover the full range of questions suggested in your blurb, which I think would be impossible in the time. So it will be narrower, and I hope, therefore, also more discussable and less general. I'm going to ask the three speakers to tell you first what they themselves do, and then I'll pick up again and sort of introduce the substance. And I'll begin with Lauren, who is at the end there, to say what she does. Many of you will know her.
So I'm Lauren Zeitels. I was a Gates Scholar in 2006, where I did an MPhil in human genetics, working on the genetics of type 1 diabetes. I then went on to do my MD PhD, my PhD in human genetics, but mostly working on microRNAs and cancer, and am now an internal medicine resident at Massachusetts General Hospital.
Liz Zhang.
Hi, my name is Liz Zhang. I first started off as a Gates Scholar in 2007, where I did an MPhil in development studies. That was during my third year of medical school. Went back to the US to complete my medical training in internal medicine, came back here for a PhD in sociology, and where there was also some ethics in it, and just graduated yesterday. And now I'm an assistant professor of medicine at University of California, San Francisco.
Michael.
Hi, everyone. My name is Michael Young. I came to Cambridge in 2011 as a Gates Scholar to study philosophy. So I completed my MPhil and then went to medical school at Harvard, and I recently completed that. And now I am starting residency in neurology at Massachusetts General Hospital.
Thank you all very much. I'm going to start by reminding us that concern about the ethical, social, economic, environmental issues raised by genetic technologies is absolutely nothing new. That's why we've had to narrow this a great deal. I first came across the topic, I realized as I was coming up this afternoon from London, nearly 40 years ago. I was in New York City. I went to a meeting of philosophers and physicians, and the topic of the day was genetic modification. That might surprise you. What it was about was genetically modified yeast. What it was about in particular was whether it was okay to just pour it down the drain when you've finished with it. It's what we would now call containment. Not an irrelevant set of questions. I don't remember anything that was raised as an ethical question of any great interest about the genetically modified yeast. But the thing that has stuck with me is a rather elderly physician saying at the end of the meeting, yes, you know, we had medical ethics when I was in medical school, which I would guess was probably the 1930s. So ever inquisitive, I asked him, and what were the main topics? And he said, oh, medical ethics, that's 3 topics. So I kept a very straight face, and I asked him what the topics were. And he said, well, there was referrals, there was confidentiality, and there was billing. Well, there has been quite a change since then, as you all know, in the way people discuss medical ethics. But I often call that story to mind because I think it's quite salutary to remember how our focus changes. I'm now going to ask each of our panelists to present for around 10 minutes, using an overhead if they want to, not if they don't want to. And I hope we'll build up a sequence that of connecting but different comments on one particular relatively recent genetic technology. Lauren, over to you.
So I want to start off by introducing the topic of CRISPR. So I'm sure many of you know what I'm talking about, maybe some of you don't, and so I'm going to give an overview of the science behind what CRISPR is. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. And it's generally in kind of the public thought to be used for genomic editing. That's not originally what it was designed for in bacteria. It was originally found in E. coli back in 1987. And in the early 2000s was discovered to have a role in antiviral defense for the bacteria. It's also not the newest, it is the newest, but it's not the only way that we can genetically modify an organism. But unlike talons or zinc fingers, which had largely been used previously, it's a lot cheaper to use CRISPR. And so that is why it's kind of generated so much interest in the scientific community and more recently kind of in the community at large because of the change in the cost and ease of use. It is thought that it can be applied much more broadly and easily. So just to kind of describe what it is, I'm going to put up a diagram. So it's comprised of two different pieces. You have your protein, which is Cas9, and then you have your RNA strand. Your protein, Cas9, acts like a scissors. It's going to cut the DNA. And your RNA is really kind of the key piece in what makes CRISPR special, because it's that RNA that recognizes the specific sequence in DNA and allows Cas9 to cut where you want it to. And because it's the RNA that makes it specific instead of the structure of the protein like in talons and zinc fingers, is what makes it so cheap to create. So when the Cas9 and the RNA bind together, they can then recognize a piece of DNA. So it's this sequence here in your RNA that is really the important piece. And when you add in a target DNA sequence of your own making is when you can start to do genomic editing. So the Cas9 will cleave the DNA in two different spots. So you get what's called a double-stranded break. Double-stranded breaks in DNA induce two different DNA repair mechanisms. And this is really key in kind of understanding kind of the larger implications of CRISPR and what might be some of the limitations of CRISPR. So you either get non-homologous end joining or you get homology-directed repair. Let's start with the non-homologous end joining. This occurs in 90% of the repair process, and it's very efficient, but it's not very accurate. So what it ends up doing is kind of just pasting the two pieces together. You end up with insertions or deletions of DNA. And it's very effective if you want to create a knockout mouse, because usually those insertions or deletions change what we call the frame in which we read the DNA. So you might recall that there are 3 base pairs of DNA for each protein. And so if you take away 4 base pairs, you now are not making the same protein anymore. And so especially in basic research science and kind of the initiation of CRISPR being used, that's been very useful in making knockout models. That's not generally as useful if you want to think about how it might be applied in humans, because there's not a lot of control over what's going on there. The other type of repair is homology-directed repair. And this is the one that's really going to take more advantage of this target DNA sequence that you put in, because in this sequence, you can put in a single base pair change. And so you can change you can then, when the DNA replicates and is fixed, you can add in that single base pair change. Unfortunately, for kind of the directed mutagenesis that creates, this only occurs in about 10% of the time. So it is really useful in that in 10% of the cases, if you're going to make a knockout mouse and you can screen every single embryo, then you can come up with 10% of the time when you can get that one base pair change and you can get exactly what you want. So CRISPR and Cas9, what has it been used for thus far? It's been really successful in kind of basic science research. So one, like I mentioned, making knockout animals, making animals with single base pair changes. They also can mutate that Cas9 so that it doesn't actually make a double-stranded break, but it will still go straight to the gene of interest. And so you can turn the genes on and off by making modifications to the protein. You can make modifications to the protein and alter kind of the epigenetics, so they can add a methylation group, and that'll also change if a protein is made or not. You can make these RNAs longer and introduce them to bind in multiple places in the genome and kind of edit multiple spots at once. All of these things are really being done in basic laboratories. But people have started to think about, can we correct diseases with this technology? And so where has that been tried? In mouse models of Duchenne's muscular dystrophy. So that is a disease of kind of muscle wasting. And there's one gene that's known to be mutated. What the Olsen lab did is they actually used more of this non-homologous end joining idea to create frame shifts, so to delete an exon that's mutated in that gene, then creates a functional enough gene, and it can correct in those mice the disease. They've used it also in correcting other single monogenetic diseases in mice. And then kind of what gets us more to medicine itself is that there's a Chinese group that actually has taken tetraploid embryos, so embryos that could never actually become people, and they've used CRISPR in those embryos to make genetic modifications. And that kind of really galvanized the field into thinking, can this actually be applied in humans? So it's successful, they can make the changes, but there's a lot of limitations in how successful it actually is. And some of those have to go back to this homology-directed repair. You're requiring that the cells are replicating. A lot of the cells in our bodies are not replicating. You're requiring that it uses this pathway. You're putting in a protein, Cas9, that's now just sticking around. You have to deliver somehow that protein and that RNA to the cells of interest. And it turns out that in their embryos, they had less than a single percentage of the cell. Well, half of the cells actually got the DNA in there, but none of them actually retained it, and it never went to all of the cells in the embryo. So you might imagine that's just a start of limitations there. But delivery is kind of going to be moving forward a big thing. People have thought about delivery into specific organs, which is maybe where it starts. So you could correct a congenital abnormality in the eye by just injecting a virus containing these pieces into the eye. There's already gene therapies kind of being introduced in that field already. You can think about taking out bone marrow cells from people and correcting diseases within the bone marrow cells and then putting them back. But kind of delivering this overall to people is not something that is really within our realm of possibility right now. So I think that's kind of hopefully will give you a basis of what the CRISPR-Cas9 can do and kind of where it's been thought about being used thus far.
Thank you, Lauren. Liz, do you want to, do you have any overheads?
No, I don't.
No, you don't. Okay. Over to you.
Great. So I'm going to build upon what Lauren just talked about and talk about some of the ethical challenges. And I think the point that Professor O'Neill made about how this actually brings about age-old issues in ethics is really interesting, because even though CRISPR-Cas9 and gene editing is such a new technology, it actually brings about age-old questions that we've had in ethics for a very long time in a very interesting and different way. And so I'm going to focus, so Lauren talked about somatic versus germline changes, and so the descriptions of bone marrow and eyes and that sort of thing is a somatic change where you would be editing the particular genes in the body, but it wouldn't be passed on to progeny and children. I think really interesting ethical issues lie in questions of genetic editing to the germline. So what I'm going to do is briefly talk about 4 different aspects of that. Firstly, ethical risks just in general, and then the age-old question in ethics about treatment versus enhancement and what that has to do with this, issues of disability and our perception of disability in society, and overall societal risks. Very briefly, I think there'll be a lot of time later to have ethical discussions amongst the whole group, and I think that's where a lot of the interesting discussion will take place. So this will be a very broad overview in general, just to sort of help frame this discussion. So firstly, we don't know very much about this technology. I mean, we're here in a place where people are very excited about CRISPR-Cas9, but we're not really sure, what its potential is. We don't know. We're not in any position to be doing germline editing right now. So actually, people are talking and fearful of some of the consequences and excited about the consequences, but they are actually very much in the future. And so just overall ethical questions of like, well, what are the risks that we aren't even aware of? At what points, and I think this actually happens a lot in technology, in science and in medicine, where the technology and science progresses at a pace faster than when we're able to think about it from an ethical and moral position. So what are our ethical obligations to future persons in editing the germline in general? What could we accidentally do wrong and unleash just by not really knowing what we're doing? I think there's been a question of how do we regulate this, and this is a question that has happened in the past, and there's been efforts within the biological community to put a hold on doing too many things with CRISPR before we know what's going on, although given the fact that science is a very international and globalized community, it's very difficult to try to control things. So for example, there have been Chinese scientists who have worked on germline editing before I think other scientists felt comfortable with doing that. And then there's also questions of our obligation to the population gene pool and what we're going to do beyond individuals and our obligation to be a steward for future populations. And so thinking about that and going back to ethical questions that have been debated for quite a long time are the issues of treatment versus enhancement. So you may have heard, there was actually just a feature or cover article in the New York Times about designer babies. I know in the US there was an NPR segment on it. People are really interested in this idea of, can we make superhumans? And I think that, this is an interesting question because we can talk about gene editing for treatments. We can talk about it in terms of changing variation in human beings, and we can talk about it in terms of enhancement. And when we think about these sort of ethical questions, it's very easy to say, okay, if there's an awful disease that's very debilitating for a human being, it makes a lot of sense to be able to use our medical technology to treat that person. But then what do we do when we have that ability to treat people and then now we're talking about, oh, well, this person may not have a desirable characteristic that is just a human variant. So for example, a short person. And at what point in the future when people are very accustomed to treating terrible diseases with CRISPR technology, then do we start saying, well, we should all be having taller children because that's better, or children that have, if the social desirability is for blonde-haired and blue-eyed children, then, at what point are we making those sort of changes, and then that becomes the norm. And then once that starts happening, then there's the question of, well, enhancements. And I think a question is also, well, what's the difference between a treatment and an enhancement? And so, for example, using the example of height, people can be short because they have a growth hormone deficiency due to a brain tumor. And so that's a treatment that one could say, okay, well, now we're going to treat that. But then if there's a person that's just short because they have two short parents, would that be considered an enhancement, or would that be treating? And how is that actually different? And what are the implications of that? So I think that there are a lot of questions that arise from gene editing that we need to think about that have implications for the way that we think about these various issues. From a societal perspective, we've seen back in the 1940s scary things happen in science with regards to eugenics, and there have been parallel questions being made about, well, at what point is this eugenics? At what point is this doing something that's just a treatment or enhancement? And this also, again, going back to the global nature of all of this, points to differentials in our ability to provide these treatments or enhancements to people. So are there going to be injustices and inequalities based on someone's ability to pay for these treatments? And so people who are able to, is this going to be a thing where the upper middle class have these enhancements and treatments, and then those who are working class and poor aren't able to afford it, and then we get divisions in society based on that? There can be disparities between countries as well, developing countries and countries that are very industrialized and well-off being able to provide these enhancements. And so we could see potential divisions. And these all sound very sort of science fiction. And so I don't think that these are things that are going to happen right now, immediately, obviously. But they're things that I think we need to think about because it's a bit of a slippery slope in some ways. And I think this actually also brings about some interesting questions that have happened that have been debated in both ethics and sociology in terms of perceptions of disability. So when you're doing things like enhancing people or changing characteristics, that's inherently making a judgment on what's better and what's worse. And so when this becomes a social norm, perhaps in the future, that we are going to be editing for certain characteristics, that in turn changes society's perception about these sort of things. And so this has already been an issue in the disability community. For example, deaf communities have been somewhat resistant sometimes to cochlear implants and that sort of thing because they feel that that's not a disability, but that it's a social construction. Society has changed has made this a disability. So when you start thinking about those sort of issues and how by having this new technology, how might that change the way we think about different things? And then if you get to the point where you're starting to think about, many years down the line, where in the event that these CRISPR technologies were the norm, would then there be a situation where people feel that by not doing it, you're actually harming your child? And so, I just think what's really interesting is that there's so many spin-offs that you can keep thinking about these different issues. And so, would a mother be, challenged because she doesn't want to enhance her child? And then there are also questions of predetermination and self-determination. So what if a child doesn't want to be enhanced? What if you're genetically modifying children because you want them to be tall, because you want them to be a basketball player, but actually that child's dream is to be a gymnast, and so that would be a disadvantage for that person. So I think this was, again, a very broad, sweeping discussion, but I think it just sort of, I just wanted to highlight all of the different challenges that may come up from this technology, and I'm going to pass it on to Michael.
Thank you. Well, as we've heard, we're now living at the crux of immense biomedical discovery that is really pushing out the boundary of what is and what will be possible to achieve through biomedical means. Notably, staggering advances in synthetic biology, of which CRISPR is one paradigmatic example, and biotechnology more generally, have given rise to technologies and techniques that are capable of potentially enhancing human functioning rather than merely treating or preventing disease. And questions that have been gestured at earlier is, can we reliably distinguish therapeutic from supertherapeutic applications, and should that question even matter? And what I'd like to focus on is how philosophy of medicine, in particular, how operative accounts of health and disease might bear on our understandings of the ethical boundaries of gene editing and other potential bioenhancements. Now, despite the ubiquity and importance of the concept of health, there is startlingly little consensus about what this concept means. The ambiguity of this concept generates corresponding ambiguity not only in political and bioethical discourse, but also in the parameters of the medical profession and the perceived derivative responsibilities of medical and scientific practitioners. This is made especially obvious and manifest in the case of bioenhancements, use of genetic engineering to modify human somatic and germline cells. The concept of health, like the concept of disease, can be understood in a variety of ways. On one account, health consists in the species' typical functioning of the organism. This position, which might be referred to as objectivism, and it seems to me is the one which is tacitly assumed by many in the medical profession, and is explicitly defended by a number of philosophers such as Edmund Pellegrino and Christopher Borse, endorses the idea that we identify instances of health and disease in the world through an entirely value-neutral and empirical process. On this account, health is defined as the absence of disease, which is ascribed to an individual if and only if that individual is able to perform all species' typical physiological functions with at least statistically normal efficiency. Objectivists do not deny that persons can and do value health, but rather maintain that the concept of health itself is value-free. To quote Christopher Borse, what makes one state healthier than another is a level of biological function, not anyone's values. The objectivist's understanding of the concept of health generates a distinctive account of health care. On this view, since health is reducible to species-typical biological functioning, the notion of caring for health is reducible to caring for merely that, caring for species-typical biological functioning. And this philosophical account gives rise to a straightforwardly restrictive stance on the place of bioenhancements in healthcare. Interventions that aim to take an individual or a population beyond what is statistically normal in relation to their reference class would fall far beyond the healing ends of medicine, so to speak, and no derivative professional obligation will devolve upon clinicians to administer such interventions. So, for instance, while a neurologist who provides a 65-year-old patient with Alzheimer's disease a prescription for a hypothetical drug that can improve her memory would be involved in an act of healthcare, the same neurologist who would provide the same prescription to an Alzheimer's-free patient of the same age who merely wishes to boost her cognitive functioning to a level that exceeds those in the reference class would not be involved in an act of healthcare and would not be fulfilling any obvious professional obligation in so doing. So this conception of health and the corresponding conception of healthcare appears to underpin many restrictive stances toward bioenhancements. Now, despite the initial appeal of the objectivist stance and its notable ubiquity, it is subject to a variety of criticisms that call into question, A, whether it is value-free as it purports to be, and B, whether it accords with ordinary intuitions about health. In particular, a range of clinical cases suggest that individuals might be unhealthy even if they're functioning at species-typical efficiency, and healthy even if they're not functioning at species-typical efficiency. So on alternative philosophical accounts, caring for health may have little to do with restoring species-typical functioning, but instead might consist in doing what is necessary to restore or to ensure the abilities of individuals to attain their own vital goals. The conceptual upshot of this sort of account is plainly more permissive toward the role of perceived bioenhancements in healthcare. If a bioenhancement would contribute to a patient's vital goals, then administering it to a patient may fall squarely within the clinician's responsibilities to care for health, even if it goes beyond what is statistically normal. This effectively shifts the focus of the debate from the question of whether clinicians ought to administer bioenhancements and whether society ought to support research that promotes bioenhancements to the question of which bioenhancements clinicians ought to administer, namely, which interventions are those which indeed contribute to patients' vital goals, which may vary from person to person or population to population. Now, the dynamics of this duet between physiology and well-being are undoubtedly very intricate, but calling attention to the foundational importance of our philosophical concept of health and regulating the contours of clinicians' and scientists' responsibilities, particularly with respect to the issue of bioenhancements, CRISPR, gene editing, synthetic biology, can, I believe, shed much light on some of these tricky issues. At a time when debates about healthcare coverage and resource distribution figure so prominently in bioethical and political dialogue, the question of what is meant by health and what is meant by disease is greatly important and yet is seldom addressed. And so my hope is that engaging directly with the philosophical issues surrounding the concept of health and calling attention to these issues may prove diagnostic and therapeutic in varied challenges that we meet in the course of such dialogues and debates.
Thank you, Michael. Well, there you have, in outline, I expect many of you were quite familiar with bits of all that. I'm going to lob one question and then ask our panelists to pose a difficult question to one another. My question is a very simple one. Do you think the use of the label genetic enhancement has been helpful across the last 15 years, or do you think it's merely been misleading?
So,
Anyone want to try that one?
I could take that one.
I think you could, yeah.
I think it has been it's been helpful in some ways, but in other ways, it's been misleading. And so it's helpful in the sense that it gives people the idea that these technologies will really be potentially capable of doing more than merely correcting disease and correcting genetic lesions, so to speak. Where it's been misleading is in, I think, three ways. One way is it can give the general population who might not be familiar with the capabilities of genetic engineering the notion that we're at a point in science where we could, if we wanted to, create designer babies, so to speak, or change or introduce a genetic feature that could directly alter the trajectory of a human being and potentially a population. Another way in which it's been misleading is, I think, in promoting the misguided yet pervasive notion that the phenotype, which is the appearance of an individual, correlates in a one-to-one fashion with a particular gene. And as we know now in genetics, most complex traits such as intelligence, such as social cognitive skills, all of these things are very complex genetic and socially informed traits that require the interaction of many different genes across not only the exome, which codes for proteins, but also introns, which code for modifying regions of the genome. And it's the picture is a lot more complex than the term genetic enhancement might let on. And so that's I think, another way in which it's been misleading. But I'm interested to hear.
Lauren, do you want to say anything on that?
Yeah. So I agree that in most cases, a lot of the things that we think about with enhancement are polygenic. But I want to bring up one example that actually, people have brought up with the idea of CRISPR, and see kind of whether you would think this is an enhancement or not. So there's a gene called PCSK9, which has been found when mutated and not to function at all in people. They have very low cholesterol levels. And there was some question initially whether having these really low cholesterol levels is harmful, because you need fat in your brain to kind of line the neurons of your brain. But people who have these mutations actually are perfectly normal, well-functioning people, and there's only a handful of them, but there's this thought that with this mutation and your cholesterol levels so low, you would have a lot of benefit, cardiac disease, right? We've seen with statins, kind of the trials show the lower you go, the better it is. So they've used CRISPR in mice to knock out, or to basically make non-functioning PCSK9, and it's not homogeneous across the mouse, it's just some of the cells, so it lowers their cholesterol to some extent. So with that, then, applying that to humans, don't have to take statins anymore. You knock it out once and have very low cholesterol. Granted, we don't understand entirely what are the other roles of this gene, but would you qualify this as an enhancement or something that's just disease preventative? I think that's something that you could conceivably see us using CRISPR for, but it's not really clear kind of where that falls in the balance.